All right, we'll see how today goes. All right, so we're going to talk about cell structure and function today, and we're going to start with talking about a lot of basic things about how cells operate, and then if we have time, we'll get into their actual structures themselves. Before we get going, I wanted to just do a little review of last time, right? So we did some general chemistry talking about ionic bonds, where an electron is transferred from one atom to the other, resulting in charged ions, which then are attracted to each other because they have opposite charges. Covalent bonds where they share electrons, and hydrogen bonds, right, where the electrons spend more time with oxygen and less time with the hydrogen ions in a molecule of water, for example, causing a relative difference in charge. They're not actual ions, but they have a mild charge that results in some interesting properties. We then talked about, we also talked about pH, right, which is how many hydrogen ions or hydroxide ions are present in a solution, because they can cause molecular mayhem, right? So they can actually get in there and disrupt bonds and cause all kinds of crazy things. And then we talked about things that are hydrophobic, meaning they don't dissolve in water because they are nonpolar, versus things that are polar or hydrophilic and do dissolve in water. All right, so I'm going to ask everybody to take a moment and bring your mask up over your nose for me. Thank you very much. All right, so then we talked about the 4 different types of organic molecules. So carbohydrates, right? We talked about simple carbohydrates like the mono and disaccharides, because their subunit is the saccharide, those sugar rings. And then the complex carbohydrates, or polysaccharides, like starch and glycogen and cellulose. Lipids, there were lots of different categories under lipids. So fats and oils are the ones we have the most direct experience with, 3 fatty acids attached to a glycerol. Right? And whether they're solid or liquid determines whether they're a fat or an oil. And then we have those phospholipids, those special lipids that have that phosphate group that makes the head polar, whereas the tails, the fatty acid tails, are nonpolar. And we're going to take a look at that today because that's what we use to make a really important part of our cell. We also talked about waxes and steroids for lipids, and then proteins, and we talked about the important thing about proteins. So their subunit is these amino acids that get linked together. And then depending on which amino acids you have and in what sequence, that's going to cause it to bend and fold into different shapes and configurations. And it's the shape of a protein that ends up determining its function. That's super, super important. Finally, we talked about nucleic acids. So we have both DNA and RNA in our cells. DNA is our genetic material. It has the code, the instructions, the secret to how to make and maintain a human being is all there in your DNA. And we also use RNA for some specific functions, which we'll talk about either today or next week. So before we move on into today's lesson, a few follow-up things from some interesting questions and comments last time. So there's a question about, they said, I've heard that if you cook olive oil too long, it gets acidic. And I was like, well, maybe, right? Because olive oil is an oil, so it's a glycerol with 3 fatty acids. If you hydrolyze it or break those bonds, split off the fatty acids, they're called fatty acids because they're acidic. So I went to Bertolli's website to learn all about olive oil. And in fact, the higher quality the oil is, the less likely some of those molecules have been hydrolyzed, and therefore the lower pH it will have. So anything that degrades the quality of the oil, processing, overcooking, or if you've ever had a bottle of cooking oil that's like been around for 10 years and you open up and you're like, ugh, right, and it's gone rancid. So that's the molecules have split apart and you have increased acidity. So I thought that was kind of interesting to find out. All right, so anytime an oil is broken down or a fat, right, it's going to split into those component parts, and the fatty acids are acidic. We also talked about, remember, I mentioned how heat changes the shape of proteins, right? And then Lauren made a great comment that pH can do that too, right? So the egg example we're all really familiar with in our lives. We've all cooked eggs. Well, hopefully you have, or you've at least seen a raw egg and a cooked egg. So you're kind of really familiar with how the proteins can change quite a bit. So I was like, gosh, what's a good example of how a change in pH leads to a change in the shape of proteins? And so the only thing I could think of, some of you might have some experience with, and probably a lot of you don't, but we'll see, is the cement mixer shot. Have you heard of a cement mixer shot? Okay, 2 people. Okay, 3. All right. So what this is you take citrus juice, usually lime juice, and Bailey's Irish Cream, which is a cream-based liqueur. So cream comes from milk. It has a lot of fat and lipids in it, but it also has proteins in it, right? So you know milk has a lot of protein. So milk products have proteins in them. And so the whole premise behind a cement mixer shot is that you have the person, you have these two separate things, and you have them put them together into their mouth and swish them around in their mouth. And what happens is that really low pH, that acidity of the lime juice, causes the proteins in the cream to denature or change shape. It curdles, right? That's why it's called a cement mixer. It's disgusting, right? So this is kind of where it's starting to curdle. And this is what you end up with, right? And that's in your mouth. So the only allure of a cement mixer as a beverage is to unwittingly get your friends to do something really disgusting. But that's the only example I can think of. Did anybody else ever think of another example of pH? Like, I mean, when they make cheese, sometimes they do this too. Any other examples of proteins and acids? Yeah.
My dad actually does this with cheese. He actually burns it on the stove.
When he burns cheese on the stove?
Yeah, I don't know if that's an acid.
Well, so cheese has a lot of protein in it. So it definitely, you know, when you change, so heat could, yeah, when you cook a cheese, if you cook it enough, yeah, you can change the shape of the proteins in it. Yeah. Yeah, I don't know. Interesting. All right. So another cool thing that I was like, oh, I should point out, proteins can also be something called antigens. So we talked about all the different things proteins can do. Antigens are like name tags or little identifiers on the surface of a cell or a virus. Remember, a virus is not technically a living thing. It's not made of cells. But here's our friend, SARS-CoV-2. I'm getting really up close and personal with this virus on a regular basis these days. And so antigens are any protein that can be used to identify this as the SARS virus. So it turns out, in fact, that if any of you have been to those mass screening tests that they're doing down at the field house for students who live in the dorms, what they are looking for in that test is the presence of this particular protein, the nucleic acid protein, which is normally found inside the virus. So those are the rapid quick tests. They're not highly accurate, but they can maybe be useful. We shall see. All right. The other type of test we have for COVID is a PCR test. So you've perhaps heard that language. So I thought now's a good time to let you know what that means. PCR is a process by which we extract the genetic material from a substance. It could be a shell, a cell, it could be a virus, it could be DNA, it could be RNA. We extract the genetic material from there, amplify it so that then we can detect it, right? So even if there's only a few viral particles present in the sample, we can usually pick it up with a PCR because we're going to amplify that result. So this happens to be the test that we use at student health services when we're evaluating people who are higher risk, who have symptoms, or are close contacts of known people with COVID. And so far this week, the positivity rate on those tests is 30 to 50% are coming back positive. So we're seeing a lot of cases. Yeah. Questions about any of this stuff so far? Questions about COVID? Yeah. Do I think we'll go online soon? That's a really good question. So let's think about it. I have to tell you, the vast majority of cases that we're seeing are in people who live off campus, right? They live in houses with 5 or 6 people. A lot of those people have girlfriends, boyfriends, right? So there's a lot of chains of transmission. If we go online, those people are still going to be living in their apartments and houses. It's not going to change very much, right? If we go online and allow students to leave the dorms or close the dorms, that would help a little bit, right? Because we would get rid of some of the student density, right? Get rid of some of the folks who are your age, who tend to have pretty vast social connections and networks. What might be the more likely thing that might drive a conversion to all online is if we run out of isolation and quarantine housing for the dorm students. Because if we can no longer house them, we certainly can't require them to attend in-person classes. So if we don't have enough housing for people, that might be it, and we're getting awfully close. So, I don't know. I hope not, but I don't know. Yeah, Damon.
When do you think all this is going to be done?
When do I think all this is going to be done? That's a great question. So, in the history of the world, right, we have a profound history with pandemics. Everything from smallpox to tuberculosis to diphtheria to Ebola, right? So, huh? Spanish flu, absolutely. The 1918 influenza that actually devastated young people. Young people died at really high rates from that flu. It was very fascinating. Thankfully, this virus doesn't seem to behave that way. So, what's the end game, right? So the answer to when is this going to be over is what's the end game? How are we going to get it under control? We're likely going to be unable to get it under control until we have a safe and effective vaccine. How long is that going to take? Anybody's guess. Typical vaccine development takes 5 to 20 years. We have done a lot to speed up development of the vaccine for this particular virus. It's been amazing. Like, you would not believe the things that are happening. Scientists from all over the world are working together. There are like 20 different vaccine candidates in development, right? This is unprecedented. We've never had this kind of mobilization of scientific resources before. Yeah. I'm not going to get into the political situation because then we'd be here all day. All right? But I will say, so what's our end game, right? So a lot of people are working really hard on a vaccine, and one of them recently entered phase 3 trials, right? I think I mentioned that. So the first one to enter phase 3 trials, which is exciting, kind of the last step. Unfortunately, it's the hardest step. Usually only about 50% of vaccine candidates make it through phase 3 trials, and they take time to do. So we may have a vaccine in the upcoming months, and we may not. The likelihood that we might have one in the next year or so is pretty good. The early indicators look encouraging. It's not for sure, right? So we're probably, so if it's maybe going to take a year till we have a vaccine, and then we have to get everybody vaccinated. Think about the supply chain issues. Think about the logistics of that, right? So we're probably going to be dealing with this for a year or two. I will tell you that if everybody wears masks, it makes it a heck of a lot easier to get through from now till then. Again, all the cases we're diagnosing were unmasked contact. Right? Good questions. Anything else about COVID? Right. City Council is meeting tonight to see whether they're going to start enforcing the mask mandate in town and whether they might pass some restrictions on bars. Yeah?
I actually did have some like distant family members that died from Spanish flu.
Did you really? Some ancestors who died from Spanish flu. Yeah, Spanish flu is a fascinating virus. This is a fascinating virus too. But you don't want things to be medically interesting. That's not usually a good sign, right? You want them to be really boring and like, oh, humdrum, we can take care of that. This one's tough, really tough. The final thing I will say about it is what we're seeing, which is what we expected to see, is almost all of the transmission is happening when people were exposed to somebody in the 1 to 2 days before they developed symptoms. Like, again, I can't even tell you how many times it's like, well, I was hanging out with my friend, and then the next day they get a sore throat and a fever, and then they got tested and they have it, right? That is the peak time of contagiousness for this virus, is right before you get symptoms, right? You feel fine. Tricky little bastard. Okay, we're gonna talk about cells. All right, cells. So, cell theory, right? So remember, in science, we have a lot of evidence to support an idea of how something works. We call it a theory, just like gravity is a theory. So we still actually call it cell theory. We're open to the idea that we could be wrong, and that there aren't cells, and that they don't behave in the way that we think they do, right? We have to, as scientists, be continually open to the possibility that we could be wrong. Not too long ago, physicians thought putting leeches on somebody was an excellent idea, right? We have to be open to the possibility that we're wrong. So nothing smaller than a cell is alive. Here is our friend Giardia, a single-celled parasite, right? That's alive, swimming around, giving you diarrhea. All right. This is the HIV virus, magnified relative to this about 1,000 times. It's teeny, tiny, right? Just a little bit of genetic material inside a protein shell. Not alive, not technically. And certainly a carbon atom is definitely not a living thing, right? That's what makes up steel. It also makes up a lot of us, but on its own, it is not a living thing. So, cell is the basic unit of life. That's the take-home point here. And so all living things are made of cells, maybe just one cell, like these little guys that you might find in pond water over here, or maybe multiple cells. If you're made of multiple cells like we are, you specialize. So we have different types of cells that do different types of things. And so next week we're going to start learning about those different types of cells and how they are tailored to perform specific functions and do specific things for us, right? Oh, this is that slide, right? So if you have multiple cells, you have lots of different ones. They specialize, okay? Even our bone is made of cells, okay? Highly specialized ones with really special stuff that they put around themselves that makes bone really hard. So it's all about what kind of cells you have. This is one of my heroes. I love the period of the Enlightenment, if you study history. So when people actually said, well, maybe there aren't four humors, maybe bleeding people isn't a good idea, let's actually do some experiments, right? So the first time, this is kind of the birth of science, the first time people said, well, let's actually do some experiments to see if what we think is a good idea actually has any evidence to back it up. So it used to be believed that life could spontaneously generate from nowhere. And one of the examples that was used to show this idea of spontaneous generation, this spontaneous manifestation of a life force, was that if you put a piece of meat out and you let it sit, after a few days there would be maggots on it. They came from nowhere, right? Spontaneous generation of living things, right? So this guy, Francesco Redi, in Italy, he was like, nah, I don't think so. So he designed an experiment. He said, all right, I'm going to put a piece of meat in a jar, and lo and behold, maggots are going to develop. But he happened to notice that there were some flies landing on the meat. So he said, aha, I'm going to put a cork in that container so the flies can't get to it. No maggots. And I said, all right. Now I'm going to put a piece of gauze over the top. And he saw the flies would land there, and then a few days later, maggots. So he was like, aha, those maggots are coming from the flies. And this may seem like, duh, to us, but at the time, this was like groundbreaking, earth-shattering, and religion-threatening idea. Right? Because they thought life could just poof, right? God would just say, let there be maggots, right? So this idea is totally new, that all cells can only come from other cells. And it's true, or at least we think it is, right? This is part of cell theory. So we're pretty sure that you can't make a living thing unless you already have a living thing to make more of the living thing. All cells come from other cells. Which then kind of makes you wonder, well, where did the first cell come from? Well, we're still working on that. Okay? All right, so he's my hero. Saw a statue of him in Florence. I was pretty excited. All right, cells are also usually really small. And the reason that they're small is their ability to interact with the environment is super duper important. And they interact with the environment via their membrane, their surface. And the bigger you are, the more volume you have per surface area. If you are smaller, you have a lot more surface area per volume. They need a lot of surface area, so they have to stay small. Okay, so most cells are microscopic. You can only see them under a microscope. So until Leeuwenhoek and others, also in the Enlightenment, invented the microscope, we didn't actually know they existed. All right, so this is a logarithmic scale. What that means is every little tick here is 10 times greater than or less than the other. So a human egg or oocyte, you can just barely make it out. It's about 1/2 millimeter in size, about the size of a little speck that you would make with a pen on a piece of paper, right? That is one of our biggest cells that we have, right? Plant cells, most animal cells are much smaller than this, right? Bacteria are smaller. Viruses are teeny, teeny. Here are our molecules and atoms. All right, so yeah, they had to look at microscopes. We don't have time to watch this, but I love SciShow and Crash Course and all those things. So if you want to go back and watch that video, it's fun. All right, how are cells organized? So three basic components that we're going to find in all cells, right? So we're going to start with a little basic structure here. Three basic components that you're going to find. One is the plasma membrane, okay? Another word for that is cell membrane, but we're going to use the word plasma membrane. Why? I don't know. The people who are the anatomy gods said we have to call it the plasma membrane. So the plasma membrane, and that's really important because it distinguishes outside from inside. It distinguishes cell from not the cell, right? You need a barrier. You need a border. Then there's cytoplasm, which is the jelly-like goo inside the cell. So all cells have a plasma membrane, cytoplasm, and then they have organelles. And that name is not by accident. It means little organs, right? So little parts of the cell, they're going to do the different jobs that keep the cell doing what it's supposed to be doing. You notice this particular cell also has a nucleus. Not all cells have nuclei, which is the plural of nucleus. Animal cells happen to have a nucleus. Plant cells happen to have a nucleus. But there are some cells and some living things that don't have a nucleus, right? Bacteria. They're called prokaryotes. We're eukaryotes. They're still cells, and they still actually have DNA, but it's just not contained in a nucleus. It's just kind of hanging out in the cell. So that's kind of interesting. Another difference between our cells and other cells, so here's an animal cell with its cell or plasma membrane. This is a plant cell. Plant cells have a cell wall. We do not have a cell wall. We do not have a cell wall that gives us that kind of rigidity and structure. That's going to be important later today when we talk. We do not have a cell wall. Our cells are relatively flimsy. Okay. All right. And that's cellulose, remember? That polysaccharide that only plants make. So your animal cell, typical animal cell, right? You're going to have a plasma membrane around the outside. It's going to be filled with cytoplasm, a whole bunch of different organelles, which we will get to, and a nucleus in the middle. All right, so that's that part. Hopefully mostly review. Now we're going to talk about this plasma membrane because it's super duper important. And you might remember or you might not what it's made out of. These little molecules here with the little heads and the little tails sticking down, those are the phospholipids that we learned about last week. So that's that phospholipid bilayer where you have some on the top and some on the bottom, and the tails are all in the middle. So what's interesting about the structure of the plasma membrane is that it is polar on the very outside, so it interacts well with water, it's hydrophilic, polar on the very inside, but in the middle, it's hydrophobic, right? It's nonpolar, it's fatty in the middle, and this is going to be important for how things can move across it. There are also some interesting accessories that we can find in the plasma membrane. Proteins, right? So we talked about how proteins can be in the plasma membrane to serve as receptors or to serve as pumps. So we're going to find proteins in the plasma membrane. We're also going to find some cholesterol. Remember we talked about that. It's a steroid. It has those carbon rings. Cholesterol is an important structural component of the plasma membrane. Helps give it some substance and some heft. We also have other little things hanging off, right? Sometimes carbohydrates, sometimes a glycoprotein, which is a fancy way of saying a carbohydrate stuck onto a protein, or a glycolipid, which is a fancy way of saying a carbohydrate stuck onto a lipid, right? So we're going to have some other things. Some of these are going to act as antigens or name tags or identifiers as well. So that's kind of the basic structure of the plasma membrane. And sometimes you'll hear it referred to as a fluid mosaic model. And it's like, what does that mean? So I want you to imagine in your head a swimming pool. And the entire surface of the swimming pool is covered with little plastic hollow balls, like from a ball pit, right? So it's totally coated. It's totally covered with little plastic balls, right? But you can get in there and you can move around, right? You can move through there, right? Fluid mosaic, right? So everything is just a little bit loosey-goosey, which is kind of weird to think about, that the stuff making up the insides of me is a little loosey-goosey. It's a little disconcerting, I have to admit. All right. So that's our plasma membrane. Now, because of that fatty tail of the phospholipid that forms the middle of the plasma membrane, anything that is polar or large cannot fit through. Right? Because you can't get through oil if you're water-based. You can't get through oil if you're polar, if you have a slight charge. So this is super important. We call it selectively permeable. It's very choosy. Only very small things that are nonpolar can move freely through that fluid mosaic. Right? So I'm going to say that again because it's kind of important. Only small, nonpolar, meaning not charged, molecules can move easily and freely through that plasma membrane. So in the case of water, for example, these are little water molecules. You'll remember water is polar. It needs a special door. We'll come back to that. It can't go through on its own. Over here, this is oxygen, carbon dioxide. They're gases, they're tiny, and they are nonpolar. They can move through that fluid mosaic, move through those balls on the pool surface, no problem. No problem, in and out, wherever they want to go. Totally easy peasy. Okay? Small, nonpolar molecules can move freely across. Everything else can't. All right, so what does the plasma membrane do? This is review. It keeps the cell intact, and it's selectively permeable. Very choosy about what we let through. Only small, nonpolar molecules. All right, so now we're going to talk about the movement of substances across the plasma membrane in general. Okay, so to set it up, we need to do a few basic definitions. Solute. A solute is something that is dissolved in a solvent. Okay? So I've got my container of fluid here, water, and I have little molecules that are dissolved in it. So those molecules are my solute, right? And we're water-based organisms, so our solvent is pretty much always water. Okay? So when we talk about the movement of solutes, we're going to talk about diffusion, facilitated diffusion, and active transport. We're going to go through these one by one. When we talk about water, we have a special name for that. We call it osmosis. So always when we start talking about these things, give yourself a moment and say, wait, what is moving? Is it a solute or is it water? Because it's going to be very different. Your answers are going to be very different depending on what we're talking about. Okay, so how things move depends on concentration. It happens to be a law of nature, second law of thermodynamics, that things want to be spread out. They don't want to be all crowded or concentrated in one place. They want to move toward less order. If we didn't have to physically distance from each other, I would make you all get up and squeeze into this corner of the room and just say, okay, now just make yourselves comfortable, and you would naturally spread out. Okay? This is a natural tendency of the physical world. Things will spread out. They don't want to be all clumped up over here. So over time, they're just going to be equally dispersed and spread out. That will happen on its own. It is passive. It doesn't take any energy. You don't have to do any work to make that happen, right? It's like letting something fall off a table, all right? Unhappy solute, very happy solute. So we talk about something called a concentration gradient, and sometimes this gives people a little bit of like, ah. So concentration gradient is this idea that if you have a high concentration of certain molecules in one place and a low concentration in the other, right? So over here I have a high concentration, over here I have low. To go from here to here, I'm going down the concentration gradient, okay? So to go from high concentration to low concentration is going down the concentration gradient. Those solutes to move to the other side, right, where it's less crowded. That wants to happen on its own. It is passive. And it's called diffusion, right? So diffusion across a cell membrane is the movement of small, non-polar molecules from an area of high concentration to an area of low concentration. If there is more oxygen outside of my cell than inside of my cell, that oxygen will move into my cell until the concentrations are equal. That takes no energy. It happens freely, right? That's what Mother Nature wants. So here's kind of what that looks like. So here's my cell membrane with my phospholipid bilayer. Bilayer just means 2 layers of phospholipids. If outside my cell I have a lot of solute, and if it is small and nonpolar, and inside of the cell I have none or very little, then over time it's just going to move in until the concentrations on the 2 sides are equal. This is diffusion. Happens all the time. It's happening right now in all of your cells. Oxygen is passively diffusing into your cells because you use it up, so the concentration goes down, so oxygen's going to move in, and carbon dioxide is constantly moving out of your cells at this moment. All right. Oh, look at that. This is how oxygen is in our cells. So I kind of already alluded to this. So if we said this is oxygen, this is the outside of the cell, this is the inside of the cell, this is carbon dioxide, we make carbon dioxide as a waste product. We need oxygen, right? So this can happen in 2 directions, right? So, sorry, it gave me a Glee flashback to New Directions. So, right, so things can move either in the cell or out of the cell. Again, down their concentration gradient from where they are more crowded or more highly concentrated to where they are less concentrated until things even out. All right. There's a problem, however, if the molecule is a bit large or if it's polar. Polar molecules can't get across the lipid tails in the plasma membrane. They just can't, right? Only small non-polar substances can make it across. So it just so happens that glucose, right, our blood sugar, this is what it looks like molecularly, and you might see those little symbols with the little pluses and little minuses. It has these hydrogen bonds in it. They give some areas of it a slight charge and some areas of it a slight, positive, some slight negative. So glucose is a charged molecule. It cannot get across the membrane by itself. So we have to help it out. We do something called facilitated diffusion. So what we do is create a special doorway with a protein channel that will allow glucose to be able to go where it wants to go. So it's again going to go down its concentration gradient. It's happening in you right now. Glucose is moving into your cells because you're using it up inside the cell, so you have more in your blood than you do inside the cell. Right? But in order for it to do that, you have to have this special doorway. So you have to have a special protein that allows that to happen. So it's still passive, doesn't require any energy, just requires this special doorway. So this is called facilitated diffusion. To facilitate something means to make it easier for it to happen. Okay, so this is facilitated diffusion. All right, so diffusion is movement of a solute down its concentration gradient from where there's more of it to where there's less of it. Therefore, it is passive. If it's small and nonpolar, it's just simple diffusion. If it's larger or polar, you need to have facilitated diffusion. You need to have somebody to let it through. Questions about that? All right. What if I want to move all this stuff out? So for example, in my cells right now, I do not want a lot of sodium. I do not. I have some very important cellular processes that depend upon, my life depends upon, there being much more sodium, much higher sodium concentration outside of my cells than inside of my cells. Mother Nature doesn't want it to be that way, so I have to do some work, right? If I want to move these guys up to where there's more concentration, I'm going to have to use energy to do that. It's going to take work. So this is called active transport. Oh. I have no idea whether that's there, right? So if I want, so say this is the inside of my cell, this is the outside. Well, no, this was the other way. It doesn't matter, right? If I want to move something against its concentration gradient and make it more crowded on one side than the other side, I'm going to need to use energy to make it happen, and I'm going to need to do work to make that happen. So in order to do that, I have a protein. So we also have protein pumps. Some proteins act as pumps. They use energy, and the cellular form of energy is ATP. You probably remember that from high school. right? So that you can actually shove something in so it's of a higher concentration in one place than another. So in our cells, we do this. This is kind of a busy diagram. Sorry about that. But inside of our cell, normal sodium concentration is 15. Potassium is between 60 and 80. But outside of our cell, sodium is normally 135 to 145, and potassium is 3.5 to 4.5. In fact, one of the quickest ways to kill somebody is to inject them with potassium. It used to be part of lethal injections. It'll stop their heart. Right? So we need to control this really carefully, and we need the concentrations to be very different on one side versus the other. So we're going to use a protein pump that's going to use ATP to move things against their concentration gradient. Super duper important. This alone, this active transport of our cells maintaining these differences in concentration of certain solutes takes, wait for it, 80% of our total energy requirement. Most of the energy that you use goes to doing this. That's how important it is. Okay? And how much we have to do it to stay alive. It's pretty crazy. All right. So we talked about diffusion, movement of solutes, passive, down their concentration gradient. Small nonpolar, they can go fine. Larger polar, you need special protein channels, so it's facilitated. Active transport, we're going to move it against its concentration gradient, get it more concentrated in one place than another. And that's going to take energy and a special protein. So if instead of text you prefer a graphic, right? So these things are passive, both simple diffusion and facilitated. Active transport would be over here. These two both require a carrier protein. Right? Let's Kahoot. Take out your phones. All right, so osmosis now, new chapter here. Osmosis is the movement of water. Let's say that again. Osmosis is the movement of water. Whenever we're talking about osmosis, we're talking about water moving. Now, water, as you remember, is polar because of its hydrogen bond. So we have to have a special channel that allows it to move through the plasma membrane. So we do. It's called aquaporin. Ah, pretty smart name. Aqua for water, pore is in an opening. Aquaporin. That is the protein channel that allows water to go through even though it is polar. So you would be tempted to call this facilitated diffusion, and you wouldn't be totally wrong, except that whenever we're talking about water, we call it osmosis. Right? So if you ever get a question about the movement of water, it's always osmosis. It's never called diffusion. All right. So the interesting thing is that sometimes you have a solute that can't get across the plasma membrane. So here, in this first situation, I have a lot of the solute over here on the left and not a lot of the solute over here on the right. If the solute could cross the membrane, it would, right? It would diffuse down its concentration gradient and even everything else out. But if it can't, this is so uncomfortable for the world and for Mother Nature and for the state of matter that water will move. It's like, fine. Then I will move over here to water this down and make it less concentrated. So the water will move from an area of low concentration of solute to an area of high concentration of solute to try to water it down. Okay? So it's kind of the opposite. It will do this against gravity. It will do this against a lot of things, and we're going to kind of talk about that because this becomes super duper important for humans and for human health. All right, so water is going to move from an area of high solute concentration to an area of low solute concentration, right? It's going to move over there, try to water those things down until the concentrations are equal. So therefore, we have a whole special series of terms that we use to describe how concentrated a solution is as compared to the concentration of the solute inside the cell. Yeah? What did the last slide say? I don't know, when I'm recording it doesn't let me back up. Let's see, water will move from an area where there's a low concentration of solute into an area where there's a high concentration of solute, right? So I'm going to try to water it down, basically, to try to even it out that way. So it's really important for us to know how concentrated a solution is compared to the concentration of those solutes inside of our cell. So that's called tonicity. How strong is it? How much tone does it have? Tonicity. So our terminology is as follows. Here's my cell. This is my solute. If there is a lower concentration of the solute outside of the cell compared to the inside of the cell, that is called hypotonic. Hypo as a prefix means less than. Okay? So hypotonic has a less of a strength, right? Lower concentration than what's inside the cell. If it has the same concentration, we use iso as the prefix because iso means the same. So if the concentration of solutes is the same both inside and outside of the cell, that is isotonic. Finally, hyper. You know hyper as a prefix. It means too much, right? So hypertonic solution, you have more solute, a higher concentration of solute outside of the cell than you do compared to inside of the cell. This is super duper important because water will move, right? So let's think about this. You know what? I'm going to show you that. Yeah, there we go. So these are some red blood cells. If I put them in a solution that is isotonic, where the concentrations are the same inside and outside the cell, a little bit of water will leave the cell, a little bit of water will move into the cell. Net movement of water will be 0. The cell will be happy, right? So that's good, right? So some will move in, some will move out, but overall the net movement of water is the same. If I take these cells and I put them into a hypertonic solution, so hypertonic, so the solution is more or less concentrated than the inside of the cell. Say it out loud. It is more, right? So there's a higher concentration outside of the cell. So where is water going to move? Into or out of the cell? It's going to move out of the cell, right? Water is going to move to the area of higher concentration. So if I take some red blood cells and I plunk them into a salty solution, right, with too much sodium in it, what's going to happen is water is going to move out of the cell to try to kind of water down that solution, and my cells are going to shrivel up. We give that a special name. We call it crenation. It comes from the word crenellations, which refers to those things like on the tops of castles, and somebody thought this looked kind of like the tops of castles. I'm like, I don't get it, but that's how they named them, okay? So crenation. So I can shrivel up somebody's cells by putting them in a hypertonic solution because water will move out via osmosis to try to water down that solution. Conversely, if I put these cells in a hypotonic solution, right? So now there is less solute outside of the cell. There's a higher concentration of solute inside the cell. Which way is the water going to move? Into the cell, absolutely. And in fact, the drive to do this is so powerful. Not only will water move into the cell, and I don't know if you can kind of appreciate the cell's going to get getting kind of big and fat here, but this will continue occurring if the difference in the concentrations is severe enough until the cells actually burst open. We call that lysis. Okay, so lysis is the bursting of a cell that occurs when you put it in a hypotonic solution because water moves into the cell because of osmosis. This is where a cell wall would have been handy. This doesn't happen to plant cells. That's why they have a cell wall. All right? Our animal cells, no cell wall, just this flimsy fluid mosaic plasma membrane. Jeepers, we just pop. It's terrible. Right? That's why you can take a flower or a piece of a plant and put it in a jar of just plain water. It'll be fine. Can't do that with us. All right. Here's another picture, right, of the same kind of thing of what will happen to the cell. So what happens if you drink a lot of water in a short period of time? Let's think about it. So first of all, a concept that you may not already be familiar with is the fact that anything that you put into your body, if you can absorb it, you will, right? So there's no such thing as like, I drank too much water and so some of it came out the other end, right? It's all going to get absorbed into your bloodstream. Yeah?
You drink a lot of water in like a short amount of time, you're not going to be running very fast.
That's true. You're not going to be running very fast. Yeah. Absolutely. So you drink that water, you absorb it because your stomach and your intestines, they don't know any better. They're going to keep absorbing it. It gets into your bloodstream. You get more and more water in your bloodstream. Your blood becomes less and less concentrated. Your blood becomes hypotonic relative to your cells. Water, therefore, moves into your cells, and your cells can burst. And so this is, if you've ever heard of water intoxication, that's what that is. And unfortunately, it's, people get this idea that water is harmless. Right? So there was a radio station actually a few years ago that did a contest called Hold Your Wee for a Wee. So they had this contest where people had to come and like drink a whole bunch of water and like not take a break and go to the bathroom. And if they won, they would win a wee, And Jennifer Strange was a single mom with a couple kids at home. She didn't have a lot of money. This was their opportunity, the only way they were going to be able to afford to get a Wii console. So she was extremely motivated and determined to win this contest. And she did. And she died. Because the cells in her brain went poof. Right? So isn't it interesting? We think, oh, it's just water. It can kill you. Right? So and unfortunately, it was really sad. There were lots of warning signs that something was really wrong, not only with Jennifer, but also some of the other contestants. They were complaining of headaches. They were complaining of nausea. These can be really ominous signs of brain swelling. And they blew it off. So, so they were found negligent and sued, but, her life was lost. So it's interesting because anything that you drink that is hypotonic can do this. So this is why a lot of times on marathon courses and stuff, they'll have Gatorade in addition to water, right? Because water is hypotonic. Gatorade is still hypotonic, but not as bad, right? So it's less likely to do this to you. Interestingly, beer is also hypotonic. So I once had a patient who is a chronic alcoholic. And interestingly, most chronic alcoholics will drink liquor because you don't need as much volume. But this guy drank beer, and he would end up in the hospital all the time because his blood got hypotonic. You'd have to bring him in to dry him out. That's where that phrase comes from. All right. So osmosis is the movement of water. Yes, Morgan. So if you're in the water for too long, why do you get all wrinkly, right? And so the next time you have an opportunity to do this, it would be great if you had like a ring or something to like check the actual diameter of your finger. Because it looks like you're shriveling up, but what's actually happening is you're swelling. So what's actually happening is the water is moving into your cells and making some of them bigger and heap up. So when you get all crinkly from being in a tub too long or swimming pool too long, you're actually getting bigger. It just looks shrivelly and weird because it's not equally distributed because of fingerprints, which are because of dermal papilla, which are pretty cool. Yeah.
Do electrolytes have an effect on the blood?
Yeah, so electrolytes are solutes. So electrolytes are things like sodium and potassium and chloride. So when you get a sports drink that says with electrolytes, right? They're telling you it's not as hypotonic as water. It's hard to get a beverage that's isotonic because our blood is actually pretty salty. Our cells are pretty salty. So if you don't like the taste of Gatorade already, you wouldn't like the taste of an isotonic bit of fluids. Okay.
Is that why sometimes they have people that are dehydrated drink Gatorade? that water?
Yes, that's why we have people who are dehydrated drink Gatorade instead of water. It's also why if I have somebody coming into the clinic or the emergency room who's dehydrated because it's a super hot day and they've been sweating a lot, or because they've been vomiting or have diarrhea or whatever it might be, all the things that can kind of dehydrate you, what kind of fluid am I going to give them in an IV? Lauren? Huh? Saline, yep, which is salt water. What kind of concentration? Hypotonic, isotonic, or hypertonic? Yeah, that's what people usually think, right? You would think, well, hypotonic. If you're down on water, we'd want to give you extra water. Turns out, most of the time, that is far too risky to attempt. So what we do instead is we give them isotonic fluid, and then we let their kidneys get rid of the extra salt. Otherwise, I might kill them by accident. Never a good thing. Right? But if I give isotonic fluid to somebody, I know I'm not going to kill their cells. Right? And then they have kidneys and stuff like that can deal with it. Do all the fine tuning. All right. So now we're going to talk about moving big things across the plasma membrane. Every now and then we have something really big. And so it's like beyond any carrier protein. So if we have something really big and we want to get it out of the cell or we want to bring it into the cell, how do we do that? So this is kind of like a whole different section. So endo as a prefix means within. Cyte, C-Y-T, means cell. So endocytosis is the process of bringing something inside of a cell, right, within the cell. So for example, here is a plasma membrane. Here's, I don't know, a little food particle or something that the cell wants inside of it. What it's going to do is basically just heap up the plasma membrane around it, form a bubble, then let the bubble break off inside. So that's endocytosis. And so if it's a solid, so there's these, the thing I care about is endocytosis. Sometimes we'll do it with liquids, and that's called penocytosis, or sometimes we'll need to have specific receptors for that, right? So endocytosis is how we just kind of like, right? We like fold it in and bring it inside. As you might imagine then, exocytosis is the opposite, right? We have a little sack of some stuff that we want to get rid of, some cellular garbage perhaps, or maybe a hormone that we're releasing, or a neurotransmitter. Neurotransmitters are released this way. We're going to take this little bubble, stick it onto the plasma membrane, then open it and release the contents outside of the cell. This is how nerve cells signal. They release their particles via exocytosis, because exo, prefix like exit, means going out of. Okay? So endocytosis and exocytosis, we like do this thing with the plasma membrane. So this is everything that we've talked about so far in terms of movement. So remember, if it's water that's moving, it's osmosis. Everything else is either diffusion, active transport, or endocytosis and exocytosis. So this is just for you to kind of review and quiz yourself with later. Let's see how well you captured what we did. A little bit now about organelles. So these are like the mini organs, the little parts inside the cell that are going to do a bunch of different jobs. You've probably learned these before in high school biology, so we're not going to spend too much time on them because it's just kind of like memorization. This does this, this does this. All right, there they all are. All right, nucleus, right? You know this as the command center of the cell. The reason that the nucleus is the command center of the cell is because it houses our DNA, which is our genetic information, that has all the instructions on how to do everything that is involved with creating and maintaining a human body, right? So the nucleus is the command center because in us, right, it houses the genetic information. So our genetic information is DNA. It's also packaged up with some proteins, so you might see the word chromatin used, right? Nucleus, right? This big thing, often in the center of the cell, but not always. Not always in the center. We'll meet some cells where it's not in the center. And so here again is a representation of a cell, the nucleus, and we can see these little X-like things, which are called chromosomes. So here's what a chromosome is. Here's our DNA, right? That's double helix, that's spiral, that spiral ladder with the A, T, G, and C's that we learned about last time. We package it up, ball it up with some proteins, and stick it into a package called a chromosome. So when somebody's talking about your chromosomes, they are talking about your DNA. They are talking about your genes. They're kind of talking about all the same thing, right? The chromosome is just how we happen to package up the DNA. Interestingly, we only do it when we're about to like make a copy of the cell. Most of the time, the DNA is just kind of hanging out and it's not in the chromosomes. But anyway, that's what a chromosome is. There we go. We also have this little dense part in the middle of the nucleus. So this whole thing is a nucleus, and this little dense dark thing in there is called the nucleolus, which is like the mini nucleus. But it doesn't house DNA. In fact, what the nucleolus does is it makes RNA. So we've talked about RNA before. It's like DNA, except it's single-stranded instead of double-stranded, uses uracil instead of thymine, so one of its 4 letters is different, right? So what do we use RNA for? We use RNA for 2 different things. The first thing that we use it for is a way to communicate the information from our DNA, because our DNA does not leave our nucleus. It stays there, right? It's like the reference books at the library that you can't check out, right? The DNA has to stay in the nucleus. So the RNA, we make mRNA or messenger or message RNA. So RNA will make little molecules of RNA that are little copies of the code in our DNA. So we'll unzip part of our DNA, make a little RNA copy, and then that little RNA copy can leave the nucleus and go out into the cell and tell it what to do. So we have messenger RNA, right? It's like our messenger, so that works. The other thing we use RNA for is to make ribosomes. We'll talk about what those are in a sec. So again, here's our DNA. We'll open it up and make a copy of one side out of RNA, right? So we'll line up, use those hydrogen bonds to figure out what the order of the different code is. And then that messenger RNA can leave the nucleus. The way that messenger RNA ends up telling the cell to do anything is actually kind of crazy when you think. The way to actually give an instruction is that this code is a code for how to build a protein. And proteins are enzymes. Proteins are structural components. Proteins are receptors. And it's the protein that do the thing. So really, all our DNA is supposed to be protein. That's it. And then because of the enzymes and other things that those proteins are, that means all the chemical reactions happen and all the functions happen to make us up. So the way that RNA gets coded into a sequence of amino acids is by a ribosome. So a ribosome is a protein factory. A ribosome is a protein factory. It's going to read mRNA, and the ribosome itself is made out of our RNA. So weird. It's like a right? So ribosomes are made out of ribosomal RNA. They read mRNA to create protein. So ribosomes are protein factories, and because the proteins that we make, that we end up having curly hair, or straight hair, or brown eyes, or blue eyes, right? Or being tall or not being tall. Or having gonads that make testosterone, or gonads that make estrogen. All right, so awesome science with protein. Outside of the nucleus, there is a membrane that close to the nucleus is studded with ribosomes, and it makes it bumpy. So that's called the rough endoplasmic reticulum. Rough endoplasmic reticulum. So because it has ribosomes on it, it makes proteins, and then the endoplasmic reticulum part gets modified. Right? So the rough endoplasmic reticulum is going to make other stuff, like phospholipids, hormones, right? So other important things that we use for our cells are made by the smooth endoplasmic reticulum. Sometimes the smooth endoplasmic reticulum also carries out certain chemical reactions for it. Like our liver cells detoxify alcohol. They're smooth endoplasmic reticulum. How the action metabolism process in your organism. So endoplasmic reticulum, you've got the protein making part, the rough part, and the smooth part does all kinds of interesting things. Golgi. Let's look at what that thing covers. The Golgi apparatus is stacked in a membrane, kind of like pancakes almost. I like to think of it as kind of like shipping and receiving. It's kind of like the UPS or kind of the Amazon of the cell, really. So it receives things, it modifies them, it packages them, and it sends them out where they're going to go. That's the Golgi. It's also going to make these little things, some of these little things, which are called lysosomes. A lysosome is a little sac with digestive enzymes inside of it. So a lysosome is a little sac with digestive enzymes inside of it. We use it to break down food particles. We use it to break down waste particles. And if I'm an immune cell, I might use it to break down some bacteria that are trying to invade my body. So lysosomes, they're like little enzymes, little destroyers of whatever needs to be destroyed. They don't have the garbage disposal stuff. Alright. Lysosomes are important. Right? For most of us, like, lysosome, that, I didn't remember learning about that in biology class. Like, why is that even important? Well, sometimes people have problems with their lysosomes, they don't work properly. So an example that happens to also be in your textbook is Tay-Sachs disease. This is a genetic disease where the lysosomes in your nervous system, in your brain and your spinal cord, don't work properly. And so they're unable to break down the waste products, the accumulated, just as part of normal daily work in the brain and spinal cord. So what happens is when they're first born, everything's fine. And then over their first year of life, they start having problems, right? They maybe start to be able to walk, but then they lose the ability to walk. They start to be able to speak, but then they lose the ability to speak. Eventually, their brain and spinal cord completely shut down. So eventually, they kind of go into a vegetative state, and then they pass away. They don't ever live past the age of 4. So this is a devastating disease. All because the lysosomes in one type of tissue in your body weren't working. All right, so that's kind of a depressing note, but we need to end. Okay, so we'll pick up next time with a few more organelles, and then we'll talk about different cell types and tissues. Okay, so remember, so the quiz is going to get posted by the end of today, hopefully. I just have a few more things to finish on it, now that I know how far we got. And so your quiz is due this Sunday night, and your first lab, your week 2 lab, is also due this Sunday night. All right? Have a good week and weekend. Please stay safe and wear your mask. Thank you so much. All right, clean off the desk.